Hey, what's up guys, this is Adi from ShastryNation, and today we're going to talk about mutations. So now that you're back from the intro, let's go ahead and get started. Okay. Mutations. Okay. So mutations are basically changes in the DNA. And so mutations occur during transcription, translation, and replication, and also during meiosis and other things like that with chromosomes and things like that, and so mutations occur to, change up the DNA, and the mutations are basically mistakes made by the enzymes in the processes of all of the above listed, topics, transcription, translation, replication, things like that. Mutations are basically mistakes made by the enzymes in those processes. Okay, so one thing I'd like to mention is mutations don't have to be bad. Okay? Yeah, so mutations don't have to be bad. In fact, without mutations, we as humans and other animals could not have evolved at all to, better fit the environment we live in. So, mutations allow us to evolve and select for good traits, things like that, and allow us to thrive in any environment in the future. Okay? So, one thing I'd like to mention before we actually get started with everything about mutations is that you should go and watch the transcription and translation video or protein synthesis video. I'll try to have it linked in the description, but you should really go and watch that one before you come here because we will be building off the topics in that. All right, and those aren't very long videos. I think they're around 15 to 20 minutes ish, I think, but I'm not totally sure. All right, so let's go ahead and get started. Okay, so what is a mutagen? So a mutagen is basically if you break down the words, muta means mutations, I guess, and then gen means like the creation of mutations. And so basically what this does is it makes mutations happen in the DNA, or at least it increases it. Okay, increases the chances of mutations. Chances of mutations. Okay, and so basically there are a couple types of mutagens. One main one is radiation. So radiation, that's why your doctor, when he's giving you an X-ray or something like that, wears the lead, I guess, vest type thing, and he also gets you to wear the lead vest wherever the X-ray isn't needed to protect against unneeded, radiation poisoning, basically. And so mutations, radiation will actually, affect the enzymes in the process of replication, transcription, things like that, and it'll cause mutations to occur. Okay, so there's radiation. There's some chemicals will actually cause mutations too. Okay, and then there's also obviously other things, okay, that aren't listed here, but these are the two main ones that you'll need to know about. All right, so let's go ahead and talk about the types of mutations. Okay, so the types of mutations. All right, so in this we'll go from the single base pair to the frameshift mutation, which we'll talk about, which is very dangerous and it's with itself, Okay, so let's get started. Okay, so substitution. Okay, substitution is basically when you basically substitute a base pair for another base, okay, or base for another base. So for example, you might have like A, T, G, C, okay, and you'll basically the mutation might be substituting the C with like a G or something like that. Now we'll talk more about how dangerous these can be and things like that, or how not dangerous they can be. But just think about it. It could be on the very high dangerous level, I guess, scale spectrum. It could also be at like it has no effect on the organism the mutation is happening. Okay, so there's missense, and so this is a form of substitution or any other type of mutation where basically the amino acid is changed. Okay, so an amino acid, and so that's what I mean by AA. So amino acid is changed. And so these could cause like devastating results. You could have cancer, things like that if the amino acid is changed. Because once the amino acid is changed, since the protein is in a 3D structure, and all the bonds between the amino acids form that 3D structure. One amino acid change could cause the whole protein's shape to change, or it can't, or it won't cause any change. It just depends on the protein and where that amino acid that's been changed is located. Okay, so then there's nonsense. So this is basically when the codon is changed from an actual codon to a stop codon. Okay, and so basically what happens is, once the stop codon hits, if you haven't watched my video on that, I suggest you to go watch it now, because a lot of the stuff we talk about after this will be dependent on that. Basically, if you remember what a stop codon does, it basically, it'll basically stop the production of proteins in the ribosome by having the ribosome dissociate and things like that. Okay, so yeah, not good, because we could have a whole sequence of amino acids that had to be made, but if they're stopped, we won't have a completed protein basically. All right, so then there's silent, and so basically this is like the opposite of this. So this is the opposite. And so basically what a silent mutation does is it actually will it doesn't change the amino acid. So no change to amino acid. No change to AA. So no change to the amino acid. And again, I mean amino acid by AA. So there's no change to the amino acid, meaning that the protein will go along as predicted, and it'll function correctly. Now, how does this happen? You might ask. Okay, so what happens is that since there's so many different combinations for codons, I forget the maths of probability, but there's a lot of combinations. That's all you need to know, basically. And so the body has to, the cells have to actually incorporate, that there's so many combinations. And since there are only like 24 amino acids, a lot of the codons will actually code for the same amino acid. So you might have like 4 or 5 amino acids being, you might have one amino acid being coded for by 4 or 5 codons. And so this reduces the amount of, mutations that occur, since they, do the same thing. They code for the same thing and they have the same effect, So this is like if everything goes wrong, this is like a fail-safe, okay, we're not going to let this happen. It's like a protection against mutations to occur. Okay, so that's what basically happens there. Alright? Alright, so then there is the deadly one, frameshift. Okay, frameshift. Alright, so basically I'll have to explain this to you, but what basically happens is that one codon, basically not a codon, nucleotide, one base is added or taken out. One second. Taken out. Okay, so one base is basically added or taken out. So we might have this sequence. Let me get another color so it'll make more sense. So we might have this in A, T, C, G, T, C, G, A, C, G, T, A, U. No, there's no U. D, N, A, T. I think that's good enough. All right. T, G, C. Okay. So basically, let's say this one is erased. Okay. So let's say this one wasn't coded for. Okay. Now this could cause devastating results because all of these will frame shift over. Okay. So basically, you'll have A, C, G, T, C, G, A, C, G, T, A, T, G, C. Okay, and instead of coding for these three and so on, you'll code for these three, which might cause a nonsense or missense, mutation for the whole gene. Okay, and this one isn't coded for. Okay, so it might cause a major, change in the protein. Okay, so this will produce different proteins. Okay, so this will basically produce different proteins, and that's not good, because if we need a protein for a certain reason, and the bonds in the protein between the amino acids and the structure of the protein determines like what the protein does, then if we change the protein's shape, we won't have a good protein to do whatever it needs to do in that situation. Okay, so this is why frame shift mutations are so deadly, because it changes the whole amino acid sequence, I guess, in the protein, and then the whole triplet codon sequences, I guess, throughout the whole mRNA molecule. Okay, so that's not good. All right. Now, one question you might be asked in your biology class is C. Okay, so we'll have these, okay? And your teacher might ask you, okay, which one might have the worst effect? Okay, so this one, or this one, okay? And so one thing you need to think about, I'll let you solve it in a second, okay? But one thing you need to think about is, okay, how many are moving over in the, how many are being changed, okay? How many frame shifts are happening if we take out one of those or add in another one of those? Okay, so I'll give you a little bit of time to think about it. Alright, if you haven't gotten your answer yet, you can go ahead and stop the video and think about it a little more, or you can look at the answer as I go through it. Okay, so obviously the most dangerous one, I think, is probably this one. Okay, so this one is the most dangerous one, okay? This one is the most dangerous, okay? And this one is the least dangerous, and this one's like in the middle. Okay, why is this one the most dangerous? So why is that one the most dangerous? Oh, well. Okay, so let's go ahead and talk about that, alright? So let's write down the same codon without the A. Okay, so we have a blank space, G, C, T, G, A, C, T, G, C, okay? Now, we don't have one here, okay? So what happens is that all of these codons, all of these bases basically will move over, so the G will move over to here, here, etc., all the way down the stream. And so basically what will happen is you'll have different proteins coding for different things, okay? Different codons coding for different amino acids, which will cause the biggest change in proteins, okay? Now, why is this one the least? Because, it only changes one amino acid, one, I guess, nucleotide, okay? Now, yes, that's still a big change, okay? But it's not going to be, it's not, there's a chance that it might be like a silent or, something like that, there's always that chance. But what happens here, you only have one amino acid change, okay? You'll have one change here, okay? Now, these are all going to be intact, and they're going to code for a correct protein, okay? So that's basically how frame shifts work. All right, so now let's go ahead and talk about, briefly about chromosomal mutations, all right? So you have, you have 44 chromosomes, okay? You have 24 chromatids, sorry, and then 44 chromatids and 22 chromosomes, autosomal chromosomes. Okay, and so these code for, everything else other than the two other ones that we'll talk about in a future video. I want to keep it, simple. Okay, so basically in meiosis, we'll talk about that next week. Okay, basically in meiosis, you'll have this process called crossing over. Okay, so you'll have two chromosomes here, that will basically exchange data to further, enhance mutations and, genetic diversity in their offspring. So basically, one thing that could happen that could cause a major issue is that, in crossing over, this part of the DNA might go and accidentally go to this part of the DNA. Now you might think, okay, how could that happen? Isn't there enzymes, things like that to protect against that? And, you have to understand that this is a 3D structure, basically, and so there are multiple ways the DNA could actually go and settle in the other chromosomes. Alright, so basically if this one goes here and we get the red one here, if we get the red one here, then we have the same DNA here, and we have the same DNA here. Okay, and that's not good, because, we just lost this whole green section of DNA that could have been important to us. This will cause a lot of, protein mutations and other things like that, and the DNA won't code correctly. And this is like the reason for a lot of diseases, things like that, and another thing might be like, a chromosome won't dislodge. So when in mitosis, we'll talk about this next week, I guess, chromosomes will actually dislodge with, with their other, will actually separate into two chromatids, and they'll go to different sides. But, what will happen in this situation is that this chromosome will actually, this whole thing will go to that side, or this side, depending on where this force is, or something like that. And this is like, the reason why people get Down syndrome and things like that, or trisomies, or in certain locations. I think Down syndrome is trisomy in 21. And so that's an issue, that's a problem. That's not great, because these things could cause a lot of problems, and, people with Down syndrome actually live shorter lives, they don't have, superb, I'm trying not to be offensive, but, they don't have, all their cognitive skills, things like that, and they usually die early, which isn't good. And so a lot of these things should be coded for, and prevented. Okay, so now these mutations are called chromosomal mutations. Okay, so I guess that's all I wanted to talk about, basically. All right, so if you need like the ultimate fast track, to learning about mutations, I would learn what a mutation actually is, what a mutagen is, which basically causes mutations, a basic overview of the types of mutations, okay, and also chromosomal mutations and how they work to like at a very basic level, okay, and also, you have to be able to distinguish which part of the frameshift is most deadly and things like that, as we did here. Okay, and so that's basically all you need to know about mutations. Alright, so if you liked the video, go ahead and hit the like button. If you haven't already subscribed, go ahead and hit the subscribe button. I make videos like this every Monday, and I just like making biology videos and things like that. I also do gaming and technology videos. I'm actually gonna come out with the 2DS review later this week. If all things go well, things like that. And yeah, I guess I'll see you guys on the next one.